Hi, I'm Stav from Weizmann Institute of Science, and I'm going to talk to you today about how we think fungi are connecting neighboring trees. So let me first break it to you. This study is going to be about forest trees and not about fruit crops, but I think the methods and the results that I'm going to show you are highly applicative to either fruit trees. And as a wise man once told me, a tree is a tree, and it's okay to talk all about them. So first of all, a little bit of an introduction, just so we all be in the same page. So mycorrhizal fungi are below ground fungi, which are connecting, which they are below ground fungi, which associate with plant roots. And the symbiosis is based on the mutual exchange of carbon and nutrients. Trees around the world can be roughly divided into two types of mycorrhizal association, either arbuscular mycorrhizal fungi or ectomycorrhizal fungi. So what you see here in the graph is the total tree biomass of the northern hemisphere, and it's divided both by gymnosperms and angiosperms, and divided into these two functional groups, either arbuscular mycorrhizal fungi or ectomycorrhizal fungi. So we can see we have everything everywhere. So arbuscular mycorrhizal fungi are actually mycorrhizal fungi which penetrate the cell, the root cells, while ectomycorrhizal fungi don't penetrate the cells but surrounds them, forming this type of mantle covering the entire root. So ectomycorrhizal fungi play an important role in forests around the globe by improving tree nutrition and water supply, as well as connecting different tree species through common mycorrhizal networks. So already 20 years ago, Simard et al. Were able to show that carbon is being transferred between neighboring trees in the forest, specifically from a tree that was in the light to a deeply shaded tree. They were able to show that this happens. They suggested that this happens through the ectomycorrhizal fungi, but they haven't completely linked it to the fungi who are transferring the carbon. In 2016, Klein et al. Showed that more than 4% of the net forest carbon assimilated is being transferred between neighboring trees. So you can see here they had this tree that was labeled in red, and they found this labeling signature in an unlabeled control plant. They also found this labeling in the mycorrhizal community and not in the saprotrophic community, suggesting that mycorrhizal fungi are transferring the carbon between neighboring trees. So there are many ways through which carbon can be transferred between trees. The first one is direct transfer through root grafts. The second one is through ectomycorrhizal fungi connecting the two trees, through bacteria, or by any other combination of all of these mechanisms. So in this study, we excluded the direct transfer by not allowing the roots to touch each other, and we focus in the transfer through ectomycorrhizal fungi. So while the main question is, do ectomycorrhizal fungi facilitate carbon sharing between trees? There are many sub-questions to this main question, like how much carbon is being transferred? When does the transfer occur? Who are the tree species transferring the carbon? And who are the fungal partners involved in this transfer? So in order to address these questions, we used a stable isotope approach where we use the C13 as a dye for transfer. And I will explain how we did that. So we had basically a pot with 3 plants, a donor tree in the middle. And this tree was separated from a recipient tree by a fine mesh, excluding the roots, but allowing the hyphae of the fungi to go through it. On the other side, we had a plant which was the control, which was separated by a full barrier. We had 8 different pots like this, and we had 4 different species combination, either pine to pine, oak to oak, pine to oak, or oak to pine. We labeled the donor tree using C13. So in nature, we have 2 stable isotopes of carbon. We have C12 and C13. Most of the atmosphere is filled with C12. So what we actually do, we label the plant with C13 carbon dioxide, and then we look for a change in the isotopic signature of the neighboring plants and within the tree compartment. So we extract tree tissues, and then we look for a change in the C13. Just to walk you briefly through the sampling scheme. So we label the plants for 3 days, and then for the next consecutive 36 days, till plant harvest, we sample the different plant tissues, including the leaves, the root, and the stems. We also measure the photosynthetic ability of the plants using an infrared gas analyzer, and we analyze the C13 in the different plant tissues and in the soil compartment using a cavity ring down spectrophotometry. So what is this cavity ring down spectroscopy? Basically, it's a machine called Picaro, and it does a lot of fancy stuff, but it basically does, it calculates the isotopic C13 ratio in the different samples. So different trees and different plant tissues, they differ in their C13 signature, and it kind of varies between -24 to -32. So anything above that would be considered as label. And this allows us to answer questions like how much carbon is being transferred and when does the transfer occur. So I'm going to show you an example for one species combination, specifically from pine to oak. So what I basically want you to see here is that as we move from green color to red color, it means that we have a label. So the donor tree leaves were actually very labeled, and to a weaker extent, the stems and the roots of the donor tree. But what was interesting was to find this labeling in the roots of the recipient plant, and moreover in the stems of this recipient, while the leaves of the recipient remained clean for the entire experiment, as well as the control plant, which was completely excluded from the microazole fungi, and it remained clean from the labeling throughout the entire experiment, suggesting that carbon-13 was indeed transferred below ground. And I'm showing you here this result for pine and oak, but we saw this across all the 4 tree combinations. So then we go back to the most interesting question. Now, who are the fungal partners transferring this carbon? And first of all, we wanted to look on the entire mycorrhizal community in our experiment. So what you see here is a rank abundance curve of the most abundant species. And in white are the ectomycorrhizal species, and in gray, these are all other saprotrophic or other type of fungi in our system. And you can see that most of the fungi we find in our systems are indeed ectomycorrhizal. In the pie chart, you can see the distribution of the different ectomycorrhizal genera in our system. And in the heat map, you can see the relative abundance of these different type of fungi on the donor plants, which we actually sequenced. So that was just a brief look at the ectomycorrhizal community in our study. But if we actually want to know if ectomycorrhizal fungi are facilitating carbon transfer among trees, we need a more accurate approach. And for that, we use stable isotope probing. So I won't dive deeply into the protocol, but what we basically do, we extract root tips, which is the area of association between the fungi and the plant. And from these root tips, we extract the DNA. We separate the DNA using ultracentrifugation, and we separate them into light and heavy fractions, where the heavy fractions are supposed to have the C14, and the light fraction would have the C12 because they differ in their density. And then we sequence separately each one of these fractions. So the species that we find in the C13 fractions are the ones that incorporated C13 into their DNA and were actually active during carbon transfer. So then we actually look for these type of graphs. You can see here in the x-axis the relative abundance of a single species, and on the y-axis you see the buoyancy density, which is equivalent to the different fractions we have. So in the yellow you can see the heavier fraction, which is supposed to have the C13, and in green you see the lighter fractions which have the C12. And what we basically look for is for these tiny yet significant peak shifts where a species has actually incorporated C13 into their DNA and has a higher relative abundance in these higher fractions. And what we saw that out of the many different sequence species that we got, these are the ASVs, it's the amplicon sequence variant. So out of these many species that we got, the ones that were actually drifting to the heavier fractions and were actually incorporating C13 into their DNA, most of them were ectomycorrhizal species. But a species can also drift to the heavier fraction if it has a lot of GC in its DNA. So we wanted a more accurate approach to make sure that these species have actually incorporated the C13 into their DNA. And we wanted to quantify the amount of C13 in their atoms. So for that, we used an independent method called UHPLC-MSMS. And I won't get into the results, but what you see here is an example of a graph. So before labeling, you can see that we have only a little bit of C13, and most of it is in the heavier fraction. While after labeling, we see a lot more of C13 atoms in the nucleobase, and all of them are drifting into the heavier fraction. So that kind of verifies the fact that these species that we found that incorporated C14 indeed incorporated C14 and not just drifted to the heavier fractions. So to summarize the species that we found a species belonging to the genus Pustellaria, Tomantella, Terfezia, and Tuber. All of them are known symbionts of both oak and pine. Regarding Pustellaria, we don't have a lot of information regarding who is the tree host. We don't know a lot about it. But it kind of verifies the idea that ectomycorrhizal fungi are involved in the transfer of carbon. And all of these are symbionts of pine and oak. So we were able to answer four questions in this study, specifically how much carbon was being transferred, when does the transfer occur, who are the species transferring the carbon, and who are the fungal partners involved. Yet a few different other questions remain unsolved, like what is the form of carbon being transferred? And we're trying to answer this using metabolomics now. How much of the net assimilated carbon is being transferred? And for this, we are now running the compartment model, calculating the different fluxes between the different plant tissues of how much carbon is being moved. And what I find most interesting is what are the ecological consequences of this carbon? What are the ecological consequences of being connected to a network? And for this, we are looking on the non-structural carbohydrates in all of the different plant tissues. And we're also applying different manipulations of shading and drought, and we check the response of the trees and the carbon transfer. So with that, I would like to finish. I would like to thank Rotem Kanovich, who did all this work with me. I want to thank the organizers of this conference and you for listening, and I will be happy to take any questions. Thank you.